Hi everyone. Hopefully you can hear me fine. Today we're going to be listening to a presentation from Farzana from GSK. Just before we start, obviously kind of usual kind of information in terms of the fact that you are muted, but please don't let that stop you asking questions. So you can use the chat feature, obviously, to write out your questions. And we also take questions at the end. So if you'd prefer to wait rather than type them out, just raise your hand and we'll deal with those at the end of the presentation. And just to also note that this is being recorded today and it'll be available in a few days' time on the Certara YouTube channel. Just as by way of introduction to Farzana, for those of you who don't know her, if you move on to the next slide, Farzana currently is working as Associate Director and Associate Fellow in PBPK Modeling at GSK. She hasn't put on here about a bit of her history. So those of you must know that she was at SIMSIP for a long period of time, particularly involved in the pediatric populations and development of them. And obviously you'll be familiar with a lot of her papers regarding that, especially around the ontogenies. And very happy to say that we worked together in Sheffield originally. We were both doing our PhD together, and then we moved over to CAPCA. But she's got a lot of experience, especially in the field of pediatrics, and that is the topic of our talk today, talking about a specific piece of work which was looking at development and application of the pediatric mechanistic kidney model. So as to not take any more time, I'll pass over to you, Farzana. Thank you for agreeing to present to us today.
Thank you, Ollie. It's a pleasure to be here. Really, I'm excited to present this work and be here with you colleagues again. So I really appreciate this time now. Okay, I think the slides are moving a little bit slower than I was expecting, but that's okay. Please bear with me. So I'm going to talk about the development and application of pediatric Mech model, which was available for quite a while in adult simulator, all of you might know. And then in the past few years, it was introduced to the pediatric simulation. Before we actually start to look at the Mech model, I'm going to give you some background about the pediatric renal clearance and the history we know about it. And then I'm going to talk about a little bit about the structure of this model, and I'm going to show you some of the examples of how this model is working and the verification of the model. Okay, so now I have I have seen both sides. I have been in the PBPK development, and I have been working in GSK for a year now, and I see the clinic side and the clinical trials. It's quite important for me now to understand how every and each of these parameters are measured and calculated. And renal clearance is one of those that is quite often measured in clinic. We have around 30% of the drugs renally cleared in the market, and so it's quite important to understand all the routes of elimination. When we have a clinical trial, the rate of drug excretion, which is the DAE to DT, is expected to be measured and divided by the plasma concentration. But because at the moment and in one at the time t, it cannot be measured at the same time, they usually look at the delta and a period of time, and then they measure the plasma concentration as a median of that interval. So they do the urine collection during a certain period of time at different times for the duration of the study, and then they measure the concentration, median concentration between the 2 points, and then they calculate the renal clearance. But when we talk to the renal physiologist, we see they report it as the Q urine, which is the rate of urine production, and the concentration in the urine. So the urine flow times the urine concentration and then divided by the plasma concentration in that period. And obviously, when we cross out the units, we are end up with the milliliter per minute renal clearance. But there are also easier ways to do this, and not always it's measured. Sometimes it's actually calculated or estimated. There are different processes and mechanisms involved in the renal clearance. One of the most important ones and the most well-known one is the GFR filtration. And there is also more recently focus of many of the studies is the active tubular secretion. There is also a mechanism of reabsorption going on, which is less understood. So these are, as you can see in this equation, there are a lot of system parameters. We have the blood flow to the kidneys, we have the unbound fraction in blood, we have the GFR, and we also have the active secretion or intrinsic clearance for the transporters involved. And all of these parameters can be subject to age-dependent changes. I'm going to discuss a little bit about the importance of this in each of these ones. These graphs here are showing the GFR, glomerular filtration rate in pediatrics. The difference between these graphs is basically showing the age range to begin from the younger age groups on the left-hand side and moving to the adolescents on the right-hand side. And the difference between the top and bottom graphs are between the populations. So another important thing that we need to bear in mind is the population when the parameters are measured based on the size. So the top one is the Japanese children, which are smaller, and the bottom one is the Dutch children that are taller and bigger in size. So the GFR, when it's estimated, we have to be aware of the size of the subjects too. Each of these lines on these graphs are showing the different models available in the literature. Different groups have reported models for estimation of GFR, and there are different ontogeny models in here. So these are some of them, actually, not all of them, but some of them. And you can see each of them are showing slightly different patterns. Maybe the three top lines are a little bit more closer to each other, but when we look at it and then we look at the size of the subjects based on the ethnicity of them, you can see the lines are crossing with each other. So what I'm trying to say here is that the GFR is an age-dependent parameter, and there are also more parameters involved, like the size in it. The next part of the equation is the active secretion, active tubular secretion. There are empirical models in the literature. This is basically reported around 20 years ago, 15, 20 years ago by Andrew Edginton. And then at the time, it was actually working for its purpose, but it's quite empirical. So you can see that the active tubular secretion in children is being calculated as the tubular secretion of a para-aminohyporic acid in children relative to adults and corrected for the fraction unbound, and then multiplied linearly by the adult value. So there are quite a lot of assumptions here. This is the simplistic approach, and that is why it is important to know more about these active mechanisms. Now the transporters are becoming more and more important. We can look at it more mechanistically, and that's really the aim of this mechanistic kidney model that we introduced to SIMSI. Mechanistic kidney model, or we call it Mech, is is developed to predict the renal clearance of the compounds based on the physical properties combined with the in vitro aspects, and also to consider the physiology of the kidney. So it will include all the parameters I talked about earlier. There will be GFR accounted for, active and passive tubular secretion, and added to that is the metabolism in the kidney tissue, and also the active and passive reabsorption in the tubules. There is also impact of the covariates, such as age, which I was talking about, sex, genetics, and many other parameters. Also, you can look when you can look at because you are looking at this mechanistically, and you can predict the concentration in different segments. In this model, I will explain later, we have 3 compartments for the cell, for the urine, and for the blood. And look at the tissue concentration in detail, and then think about the toxicity of the drugs, which is quite important, especially in children. And there is and there is also the DDI liability can be predicted with these models, which wasn't able 15 years ago with empirical models. Now, in this Mech model, the nephron is divided into 8 segments, and that's based on the known physiology of the nephrons. So we have the glomerulus, which is the number 1, and then the proximal tubules are divided into 3 sections, and that is where most of the enzymes and transporters are located in the cortex. They are located in the cortex of the kidney. And then there will be the Henle loop, followed by the 1 compartment for the distal tubule, and the collecting duct is divided into 2 segments. And this is following the physiology of the nephron. And that is projected in this compartmental model here. We have the renal blood, we have the renal mass or the cell, and then we have the urine compartment. The blood flow is coming from the cardiac output, is the fraction of cardiac output, and then we have 2 shunts in this model. One is just before the glomerulus, and then one after the proximal tubules. The rate of the blood flow to the kidney is going to define by equal to the GFR for the first segment. And then it's moving from one compartment to another one from the blood, and then we have the clearance, passive and active clearances in the proximal tubule, and then we are moving the urine from one compartment to another. The stars I've made here, this figure is from Sibylla's book chapter. Now we can call it confidently 10 years ago. But what I've marked here, really, the stars are showing all the age-related parameters that we have in this model. So the blood flow is a function of cardiac output, fraction of cardiac output, and the fraction of the renal blood flow that goes to the kidney is also changing with age. For some of the tissues like liver, it's fixed, but the fraction blood flow is changing with age for kidney. We have the renal weight, kidney weight or renal mass is changing with age. We have the transporters, the transporter abundances are changing, so the efflux and both on the apical and basolateral parts. The transporter abundances are changing and all the scaling associated with that. We have the GFR changing with age. And we also have the metabolic clearance accounted for, which is also age dependent. So you can see how in detailed and mechanistic this model is. Also, although there are still some physiology parameters that we need more confidence in them, but I think the model is as good as it can be for now. Okay, so I'm going to talk about a little bit how these parameters are scaled in the terms of physiology, and also highlight some of the gaps here. So we know that the dimensions of the tubules will change, and there are some data for some of the segments. not for all of them. The data that is used here is based on the proximal tubule, and it's assumed that the same relationship will apply to all other segments until the data become available for other segments. And they are all, well, we had the data, so we used the adult level, which we knew, and then we had the observed data in pediatrics, so the relationship was developed and allometric scale of 0.6 was estimated. And the kidney weight has always been in simulator for a long time, and the kidney weight is a function of body weight. Okay, so the age-related changes in cortex and medulla ratio is included. The cortex to medulla ratio is quite important because we know that the function of them is different, and then the cortex is quite important because all the transporters and everything is in there. We will talk about it a little bit in future slides. But it apparently the cortex to medulla ratio, the ratio of these two segments are changing with age too, and there were some limited data on that, and that was also we tried to fit an ontogeny model to show the age-related changes in this ratio. The adult value is 0.92, and this is a fraction of that. Urine and blood flow to the kidneys are also changing with age. Okay, I think we have a question. Olli, should we address the question now, or shall we wait until later?
It's totally up to you. I was happy to do it at the end, but if you
we can do it now. Let's just talk about it now. I just need to find the question in the chat. When you say pediatrics,
yeah.
Okay, does this include all preterms? Well, unfortunately, at this stage, the model is not extended to the preterm population, but I think the pediatric team in the future will do that. Maybe Olli or one of your colleagues from the pediatric can comment on any plans for the preterm.
I'll let Trevor or Roz take that.
There's no plans for the. Sorry, can you hear me?
Yes.
Yeah.
There's no plans for preterm yet. It's availability of data is the issue. So if somebody comes to us with the data and we can do it, then fine. But until that point, with no plans to include the preterm.
Thanks, Trevor. Yes, so we are going down to from the birth to 25 years with this model at present. Okay. The urine flow that I showed in the first segment is in the proximal tubule is based on the GFR, and the GFR is different based on the sex. For boys and girls is different, and I think you're aware that in SimSip up to 15 years there is an equation that is based on the body surface area for the GFR, and that's why I showed you the slides in the Japanese and Dutch to show you that the body surface area will be different. So we are expecting that the GFR will be different in children from different ethnic groups too. And then after 15 years, the Cockcroft and Gault equation is used, which is based on the serum creatinine. And it also accounts for all the variability and everything for that. And then in each segment, from moving from one segment to another segment, the urine flow is different. But so we know that we will see a different flow with age, but there are certain reabsorptions fixed and assumed between the adults and pediatrics. And that's based on the flow from the top to the bottom. For example, at the GFR level, at the glomerulus level, we say the flow is 125. And further down, for example, before the Henle loop, the flow in the adults will be 45 milliliter per minute. So the fraction that is reabsorbed between these two segments is assumed to be fixed between the adults and pediatrics. And based on that assumption, we are calculating the urine flow in the urine segments from one to another one. And at the end, the urine flow in bladder is set as the adult value, which is 1 milliliter per minute. This value can be between 1 to 2 milliliter, but in the adults at the moment it's 1, and it's assumed the same in the pediatrics too. This value can be a little bit lower at some points or can be higher based on the co-medication or the physiological condition, diseases and everything. You can change these, and you can change the fractions as well, instead of assuming fixed fractions. And the renal blood flow, I also mentioned, it's a fraction of cardiac output, and this fraction will change with age. It's not a fixed one. Okay, now moving on to I think this is all about this. No, okay. Physiology until the ontogeny of transporters and enzymes, and that's what makes this model quite unique. So the age-related ontogeny of transporters and enzymes are included. And enzymes, unfortunately, there isn't much data on the kidney itself. So the ontogeny is assumed to be the same as the hepatic ontogeny for now, until we know a little bit more about it. But as you know, it's all open and the users can change it. In terms of the ontogeny of transporters, there are more data becoming available. When we developed the model and then when we published this paper, there were three reports of the ontogeny, and I'm going to show you some of these ontogeny models in the next few slides. Okay, this is the first one. The first paper became available is from the Chung et al., Kathy Chung, and it's showing basically the protein abundance and the mRNA activity for different transporters. They looked at a large data set from preterm until adulthood, and then they developed some of the functions here too and reported that. I'm showing just few of these. There were quite a lot of comprehensive list of transporters, but these are more relevant to our compounds. So I'm focusing on these compounds mainly. So it's in summary, they show that there are a lot of discussions in the paper, but in summary, they show that the transporters here, PGP, OAT1, OAT3, and OCT2 are changing with age, and they are increasing. Some of them are not changing that much, and some of them also show a little bit of difference between the mRNA expression and the protein abundance. What we usually use is the abundance, unless we need to focus on the mRNA, just because to make sure the correlation between them is accurate, or the activity. So at the moment in the model, we are assuming there is age-related changes for these transporters. The next set of data that was available was by the Lee et al. Prasad group, and they showed quite different data from the previous set. They also looked at the proteomic samples. They had matched samples for the liver and kidney, which is a big advantage. But in contrast with the previous study, they didn't show many age-related relationships. However, they were looking at older age groups. They are looking at the over 1 year old. The previous one was from the neonates and preterm. And they also showed different patterns for different transporters. So the aim of this paper that we were looking was to look at these transporter ontogenies in specific, combine it with all the physiology data that we have on the kidney, and then look at some verification of the model, use different compounds that are renally cleared, substrate of transporters. And then because we had some gaps like the transporter ontogeny, we did some sensitivity analysis on them. The workflow was a standard workflow basically for development and verification of the model. We used drug data focused on cimetidine, ciprofloxacin, metformin, tenofovir, and zidovudine, which are all substrates of renal transporters. And human adult physiology was used. The development many of these compounds were already available in the simulator. At the time, tenofovir wasn't available and ciprofloxacin wasn't as mechanistic. So Trevor specifically worked a lot on the ciprofloxacin to make it more mechanistic, and then verified it in adults. Now they are all available in the simulator or in the compound repository. Then the refinement part happened like any other standard workflow. Then we moved to the pediatrics. It's some sensitivity analysis on the ontogeny of transporters, and then verification happened. Just wanted to show you an overview of the studies that we used. If you look at this graph, you will see our 5 compounds and the number of the studies that was used in different age groups. So for the adults, there is a comprehensive data set that we used. Some of these were already compounds that were verified in the simulator, and we looked at them again in Mech. For something like ciprofloxacin, Mech was developed. We had more data available in children, and some of the studies were using larger age range, for example, 1 to 13, and that's why we are crossing between the two sections here, between infant and children. And then we had only the neonatal data on full-term neonates in tenofovir and zidovudin. The size of these bubbles are showing the size of the studies. So the bigger bubbles are showing the studies were bigger in size. Another point that I wanted to make is that the limited data that is in infants and neonates. So you can see there are a lot more data available in children, but we need more studies to be done in neonates and infants, more data to be able to for different compounds to be able to look at the functionality of the model too. So these compounds that we looked at were substrates of 081, tenofovir and zidovudin, 083, ciprofloxacin and cimetidine, OCT2 for metformin and cimetidine. Tenofovir was an MRP4, and metformin, cimetidine were also substrates of MATE1 and MATE2. Now we talked about the transporters, and we know that these models are showing differences. They are included in the Mech model, but by default we use the Cheong data in the simulator, and then we did the simulations. But when the other data set became available, Lee et al. Showing different trend, one of the aims of the paper was to look at differences between the reported ontogenies. So for example, for the 081 and 083, which were the transporters involved in for many of our more of our compounds, we see that the Lee et al. Are showing no difference in the age, and Cheong et al. Are showing age-related changes and quite a significant difference at the beginning in neonatal. And meanwhile, another data set became available from Cinzi Crestia, which published an in vivo ontogeny of transporters this time, unlike the other ones with the proteomic and in vitro data. So what Cinzi showed was for amoxicillin and clavulanic acid, and then she deconvoluted and back calculated the impact of GFR from the transporters. One of them was the transporter substrate. Amoxicillin was a substrate of amoxicillin and clavulanic acid, and clavulanic acid is only the GFR excreted. So she deconvoluted and removed the impact of GFR from the compounds, came up with the dotted line here. Came up with the solid line here that is showing the ontogeny of 081 and 3 combined together. So we can see that what Cinzi shows is different from what is Kathy Cheong shows, and it's also different from the Lee et al. Data. So there is quite a lot of uncertainty in this area, which we hope that in the next few years we know more about. And that's why it was important to do a sensitivity analysis on the ontogeny of transporters. So this is the example of the ciprofloxacin. I'm showing you some of the results and verification of the data here. First of all, I want to draw your attention to the age groups. We have the younger age group between 0.3 to 7 years from one study, and we have the 1 to 5 years and 5 to 14 weeks. And what Trevor did here is looking at the two sets of ontogeny that was available at the time, Cheong et al. And Lee et al. And one of them is showing age-related, especially in the younger age group. If we look at it, we see it may be better. And then the Lee et al., which shows no age dependency. Overall, what Trevor concluded from this case example was that the no ontogeny, or assuming the adult level in the verification of the model from the beginning, from birth, gives us better results compared to the age dependency and lower expression. So this is something that we hope to have clarification, hopefully with the further data that becomes available. So that's the OAT3 ontogeny and sensitivity analysis. You will see in the next slide that he did some further sensitivity analysis, and assuming there is no ontogeny for OAT3, and also looking at the ontogeny of 1A2. So the ontogeny of 1A2 was also debatable. So we have ontogeny for from the in vitro, and we also have it from in vivo. But the aim of this sensitivity analysis was really as part of the model development. So when Trevor looked at this, the file was looking at the IV clearance, and he made it more mechanistic to use it for the Mech model. And for that, he had to parameterize the model for enzyme kinetics, and that information wasn't available. So there were some reports of the contribution of 1A2, but there was no actual references to that reports for this. So Trevor did some sensitivity analysis, and the results of sensitivity analysis was that using 1A2 seems to be a good approach, and also including the ontogeny of 1A2, which is the in vivo ontogeny of 1A2, also shows to have better predictions compared to no ontogeny. So I think we are good on that. This is just to show you, because I mentioned that the file was developed again for the Mech purpose. I'm including this slide to show you the verification of the ciprofloxacin after the model was redeveloped for Mech model to include the transporters and enzyme kinetic part, and also some verification of the drug-drug interaction with probenecid, which also seems to be captured well. Okay, I think that's the ciprofloxacin case example. Then we have the cimetidine. We have different routes here. We have the IV and we have the oral cimetidine. And unfortunately, we don't have any infants or neonate data here, but we only have the older children. The model seems to be doing reasonably well here. The next example is for the metformin. We have, again, children data here, but the interesting part about it is that we have data in girls from metformin in the age of 9 to 10 years, the three graphs, and we also have the obese children that metformin is administered to. So in this case, we don't have the obese pediatric population. I hope that we have it in the near future, but at the moment we don't have. So what we did here was that we created the obese pediatric population by trying to change the body weight of the body weight function in the simulator to recreate the body size of these children. So we had the same height of the kids, but we had obese children. And then the model was verified the metformin model was tested and verified in the adult obese around 110 kilogram subjects. I'm not showing the adults verification, but then we moved on to the pediatric obese children. And you can see that we have good predictions here too. Although Hi, this is Ranga here from Chinook.
had a quick question on this plot you have here. On panel number C, you see that the concentrations for metformin go all the way up to 10 milligrams per liter. This is even the median concentrations are much higher compared to panel number A. What's different between these two simulations?
These are individuals. These are different studies, and some of them, I don't know which one to be honest. We did this simulation based on the population. The other one is one individual only. But I think this one is one individual subject, the panel C.
Okay.
And the data is from one single subject, but I think the panel B was a population. That's the difference between them.
All right, thank you.
Okay. I'm trying to move to the next slide. Okay. The next example is the tenofovir. This is one of my favorite ones because we have neonatal data here. And the other thing that we had here was, I think it was voted in the last consortium that everybody is interested to have the trial design with the body weight. And that was one of the challenges we had when we did this study. So we had the body weight of the subjects, and we had to do the body weight matched simulation. So there is a workaround for that with this custom trial design. And that takes a long time to do that. But now luckily we will get it in, I don't know, version 24, hopefully. So just not diverting from the main issue, but yeah, we have the neonatal data and we have the children data, which is a bonus here. We have the multiple dose in the pediatrics. We can see in the bottom panel, we have good predictions. We have very, very young neonates. They were just born to HIV infected mothers. And we also have the 8-hour neonates. So the predictions are acceptable. I don't know, if you ask me, I don't know why the observed data is showing different, basically time dependency between the first dose and the last dose here in the panel F. It's actually quite an interesting one for me. It might be because the subjects are very, very young, and then there are hemodynamic changes just after birth. So I don't know, happening, and in the next 4 or 5 days, it's quite established. It's quite an interesting one, but I don't have a very good answer. I would like to know your thoughts, actually. If you have any ideas, let me know too. Anyway, whatever it is, the model is not capturing the first dose. But when the neonates are a bit older, then it's getting better. Okay, the next example is, I think, our last one, and it's zidovudine. We have children from wider age groups. We have IV and oral studies, which is again great. And we have the younger ones. We have the neonates, and we have the 7-day-olds. We have 14-day-olds, 28-day-olds, quite a lot of data, which is really great. And okay, so we have the transporters included. We have the ontogeny there. We have the ontogeny of UGT2B7, and they all seem to be working together. But yeah, overall, I think it's a quite comprehensive set of data. I wish we could have the same for other compounds too. And now here I'm showing you some of the examples of the PK parameters for all of these compounds. Not only we looked at the plasma concentration time, we are also looking at the clearance Cmax and AUC, showing we are aiming that every symbol to be closer to the solid line here, which is the ratio of the observed to the predicted. And the shaded line is the 2-fold difference, and the dotted lines are the 1.5-fold. So ideally we want to be around the solid line, but what we see here, most of the cases we are in the shaded area, which shows that the predictions are acceptable. Okay, overall, this was the case examples I wanted to share with you. We have the ontogeny of renal transporters, which is great. And now we are able to use it in the PBPK model. And that's why we can use Mech-Chem now. We can predict DDI, we can predict the renal tissue concentration and toxicity. And we have included all the age-related changes that we could. So it is the best it can be at this time, but it can improve hopefully when the new data becomes available. And the examples I showed here were showing that including a correct ontogeny is important. So one of the pieces of information that we hope to have in the future is the ontogeny of these transporters. We have 3 at the moment. It seems that the one with no ontogeny works a little bit better than the other ones, but it is still to be discussed and verified more. The sensitivity analysis was done. I think this is a quite important part of developing the PBPK models, and especially it's very useful on where we have uncertainty on the data, and it's a good practice to include it. And what we didn't include in the model is the age-related changes on the permeability or passive diffusion, and it is believed not to be changing with age, but we also don't have any evidence on that, and that's why I didn't talk about it at all on the passive diffusion. And we could use more compounds for the verification, and also because these are the substrates for transporters, we hope to be able to use these compounds for verification of other aspects, including the oral absorption. Overall, the predicted and observed concentration time profile seem to be predicting well for the compounds that we have looked. It's a complicated model. I hope you appreciate the level of complexity and the data and physiology data included in this model. And it's really nice to see it is working. We need more diverse range of compounds in terms of the physical chemical properties and the range of transporter substrates to be able to have more confidence in these, and then the profiles that we have. We see that there are different patterns, but they seem to be changing with age. For some of them are not. So we'll see, and we look forward to the new research. I think that's my last slide. I just also wanted to thank my co-authors and supervisor Trevor at the time, and Ben, who was also the co-author on this paper. There was a great support from the pediatric team of 2020 to 2020. Khaled, Shian, Amita, Roz. I know the team has expanded a lot since I left SimSip, but that's the small team that we had in 2020. Okay, so Ollie, I'm done with the slides. I'm happy to take any questions.
Okay, thanks. One question that came in on the chat was about how these drugs are anti-infective drugs, including anti-HIV. Are these data from healthy or infected patients? And if infected, did you need to consider any physiological parameter changes due to the disease?
Okay, that's a very good question. The tenofovir and zidovudine, obviously the mothers were HIV infected. They were positive and they were infected. I know there's ongoing work from Khaled's team for verification of the pregnancy model, and these are some of the compounds they are looking at or published already. For the children, they were the neonates. They are by default they are going to the treatment after birth. They are, I think that's a default, if I'm not mistaken. So they are likely to be positive, but we don't have any information on the pharmacokinetic changes for the HIV positive and not infected. They were not symptomatic, that's what I know. Even in the adults, when the compound falls were developed, for zidovudin, for example, the data, most of the data is coming from the HIV positive, but not symptomatic. I think one of the parameters that was quite challenging was the clearance value. If I'm not, is Sibella on the call, because she's the expert one on that. So I think overall the difference, physiology difference, is not well established even in adults for the positive HIV positive subjects. Ali, I think it's your area of expertise too, so you can comment too. But definitely for the pediatrics, it was not considered.
Thanks, Rosanna. I'll just move over to some other people who got their hands up. Lee, did you want to unmute?
Yeah, thank you. Great presentation. I have a question on compounds that have concentration-dependent plasma protein binding, and how Mech model handles FU-GFR, and how is the volume change associated with the concentration-dependent FU?
Okay, none of my compounds were concentration dependent, but we have a feature that is allowing the concentration FU on the. I don't have the simulator open, but Oli, if you can open the simulator, perhaps you can show it. I can stop sharing here. We have on the we have on the PhysChem tab. There is an option for the FU that allows you to input the fraction unbound changes. It's opening a table, basically a grid table, and then it allows you to input the fraction unbound at different concentrations. So if you have the measured data, you should be able to use it. However, I didn't try to see if it works with Mech or not. It should.
So it will calculate the FU-GFR based on your input parameters, and how's that impact the volume? Is it also depends on like all the concentration change? It will just dynamically calculate the volume and FU-GFR based on the concentration in the plasma.
I think Sibella, I wanted to answer that question, but I just saw Sibella raised her hand. Sibella, would you like to go ahead? Then I will add.
You're welcome to start, but actually what is picked up there is exactly the problem, right? So for the transporter models, you need to have the full PK method 3, and that is not something that can easily be combined with concentration dependent FU. So you can't combine it. If anyone comes up to the great idea and a lot of nice little equations to account for that, hey, we are more than happy to add that in, but that is not as straightforward, because exactly what you mentioned, you are changing then things like volume of distribution, and the volume of distribution in transporters is a can of worms for itself, right? The VSS, we have it at steady state, it's a VSS, right? And when you change it then with the free fraction, it's a can of worms. So let's put one by one. You can use this concentration dependent FU, but under very specific circumstances. And it's not something that we have combined with the mechanistic kidney model.
Thanks, Sibella. So it doesn't
I don't
work.
think anyone has in their right mind done that until now, but hey, computer science is moving on and there will be a solution for it. Right.
Thank you very much.
And I don't think we are too far away from it. The
So
other
at least
thing,
we know there is expectation.
There are expectations. Hey, I have the expectation of Masood. I have it since probably we started at SimSub like 15 years ago, but it's just not realistic. And also we had more ideas to put even more in the mechanistic kidney model, but the computer capacities at that time, our lovely little laptop, they simply couldn't handle it. So we have cut down the Mech model quite a lot from the beginning. So as Fasana said, we have the transporter in the proximate tubular cells 1, 2, 3. That does not mean that there are no transporters in the other parts of the nephron. They are, but they are not that relevant for active transported drugs. If we are talking about other compounds, obviously, in the kidney, there are reabsorption of a lot of the endogenous compounds, also in other parts of the kidney nephron. So That model was specifically done as it was voted for, not for a diuretic compound, not for endogenous compounds. It was made for drug-drug interactions with the key kidney transporters OCT2, OAT1, OAT3. So we just need to keep that in mind, but we had already in mind that we can extend it. And I see that there is actually in future still extension on these models. And I think Fasana did a great job here presenting what can actually be done on the pediatric side. And with that, I'll give it back to you, Fasana.
Thanks. If I answer the questions like that, I definitely lose my job, but I know Sibella has.
Thank you.
Sibella is Sibella. Okay. Thank you, Sibella.
You're welcome.
We all like you.
I know, to a particular degree. Okay, let's move on.
Okay.
Were there any more questions? Yeah, I can't see any. I might have missed
So
something.
this is Ranga here. So I have
sorry.
another question here. So, and this is regarding a more broader perspective. So we are talking about ontogeny specifically in the kidney space. Now, have we looked at it when we compared and when we are looking at disposition of molecules that are mediated by both CYPs and transporters? I think you had this one example where you looked at 1A2, and it was also a substrate for OATs. And we were trying to do the comparison in terms of predicted versus observed. Have you looked at in terms of, from an oxidative enzyme perspective, how does that ontogeny correlate with transporter ontogeny, and probably which plays a major role? Because that may probably help in understanding a little better in terms of predictability of some of these molecules that could be mediated by both multiple pathways.
I think that's a great comment, let's say, because I don't know the answer to your question. I think it's quite important, but
it is something for you to consider because you've already done the work. So something for you to, because SIMCYP has the framework for ontogeny for oxidative enzymes. And since you have the data and everything, you can certainly look at it from that perspective and really try to understand what could, because there are some examples, if I could, from what you showed us, where the predictions could have been further improved. I mean, these are pediatric data, so granted, these are very difficult to obtain, no doubt about it. But if we do want to better understand some of the predictabilities, especially if the compound has a narrow window for therapeutic window, these things may be more helpful. Something to consider.
Yeah, I really appreciate your comment. That's correct, and I agree with you with every single word you said. In this case, the focus was on the substrates of the transporters, so mainly excreted or renally cleared, less on the metabolism. But we cannot escape from the little metabolism, really, that we have. And I understand your point, and I agree with it. It's just that we had, we can't find so many compounds, and then we have to live with the uncertainty that we have some enzymes here that were involved and perhaps didn't do as expected. We tried to focus on the transporters though.
Thanks. There's a couple of questions in the chat. So one, they're both regarding renal impairment, really. So first one is about chronic
Aren't they
kidney...
for you, Ollie?
No.
You're the population expert. Okay, go ahead. You and Trevor. Okay.
Yeah. So regarding the pathophysiological changes occurring in pediatrics with chronic kidney disease, like hematocrit albumin, are these changes the same as in adult chronic kidney disease patients, or do you have insight into that?
Absolutely no. I have absolutely no idea. In pediatrics, I think there was a time that Trevor and I were interested to look into the renal impairment in children, but there were no data that we could find to develop a population like that. Trevor, would you like to comment on that?
It's an interesting question, and I guess my answer would be you would expect some overlap between the adult renally impaired populations in terms of physiological change, but then you've got to bear in mind that the main cause of, I believe, the main cause of renal dysfunction in children is hemolytic uremic syndrome or nephrotic syndrome. So they're sort of specific pediatric diseases. So you'd have to look specifically at the physiological changes you saw in those, in the pediatric causes of renal disease compared to adult. But I would guess that there would be some overlap between the changes we put in the adult. Okay.
Thank you.
Thanks, Trevor. And the second question is probably more about this application of the Mech model in pediatrics and trying to use it with renal impairment. So it's essentially about how could you account for reductions in tubular secretion with compounds with renal impairment? Would that be possible?
I don't know. I didn't try that. There are different aspects to that. If the tissue weight is changing, then we can. If the renal blood flow is changing, we can account for that. If the transporters are reduced or increased, we can account for that. If the urine flow or GFR is changing, we can account for that too. So all in all, I think we can change a lot of physiology, but we need to verify it first to make sure that it is all these combinations or all these changes we make are predicting well for the disease population. That's what I think. But I think practically it is feasible.
Yeah, just to add on to your comment there, Fazana, I'm not sure anybody's really done this in the adult population yet. So I'd like to see it, as somebody, modified the Mech model for a renally impaired adult population. I think that I would like to see. I may be wrong, and somebody may have done it, and somebody can correct me if that's the case. But I think that's what needs to happen first. That we use the Mech model in the adult renally impaired populations and get it working there, and then we might try something in pediatrics, I guess.
Thanks, Trevor.
Can I add on that one, Trevor? Because that's exactly the point. I mean, we are obviously getting asked quite a lot about the renal impairment and if that is also the mechanistic kidney has been extended for that. And I think we are having there the same problem as with the cancer population. It doesn't really exist, right? There are different types of cancer. And also your renal impairment can have different sources. If you have a change really that your kidney is reduced in size and everything equally is reduced in size, you can account for that. But that might be another reason for the renal impairment than that you have lost your nephrons, but the kidney size is the same. So the assumptions you make for the source of your renal impairment is reflected in the model. And therefore, it really depends on what disease state and also what disease form we are talking about. Just renal impairment, the model offers it. So as Fazana said completely correctly, there should be all the physiology data in that a user can change it. But it should be matching to the renal impairment and the status we are talking about. And that is in itself a problem to do. And it's the same as we have with a hepatic impairment, right? Which stage is it? What do you have? And now we are talking just about the kidney and how the kidney is changing. And we know that even then, renal impairment has an impact on the other organs. It's not that simple, and find the right case studies for that. So there is definitely a lot of research still possible. The model is there. We are looking for actually good research here that people are doing it.
Yes, that's great. Thank you, Sibella. I think that was a very comprehensive way to say this. But just if it's the nephrons are reduced, we can use the macrosomal protein to account for it. Am I right?
Yeah, actually, you can even
Yes.
use the nephrons directly if you want to do that as well.
Good,
Right?
great.
That's all fit in each other because you have the number of nephrons per the million of nephrons that is linked actually to the kidney weight at the moment.
Yes.
But that relationship is from a healthy kidney. So if that relationship is different for yours, you can actually adjust that and therefore reduce the number of nephrons. And with that one, you immediately have an impact on the surface area as well as on the number of transporters because you propagate that with the proximal tubular cells per gram of kidney through. So it is all interlinked with each other there. And that was actually one of the first things that was also done in the FDA paper from Sue. So where they tried to reduce the number of proximal tubular cells per gram of kidney, which is then obviously linked with each other.
Great, thank you.
Thanks everyone for their contributions to that session, and particularly to Fazana for your talk. It was a great presentation, and it was, had a great discussion at the end there and raised some good points and good things going forward, and more challenges to go. Do you mind just putting up the final slide?
Yes,
for me?
sure, yes.
Thanks, appreciate it.
Okay.
So after the Christmas break, now the webinars are coming thick and fast, so it's not long. You have to wait till the next one. This one actually has slightly different registration, so it will be on the Certara wide registration. And just to flag that, it will be a talk discussing the NDA submission. And it's the number of trials actually is quite interesting, which were waived in this drug product label. So please join us for that on the 7th of February. You can register online. Okay, thank you everyone again, and thanks finally to Fazana, and see you next time.
Thank you. Take care, everyone.
Take care. Goodbye.